wabarakatuh and salam sejahtera. My name is Nur Aminah binti Muhammad Agus and my matrix number is S55684. So I am a student of Marine Science and I'm going to present the diversity of marine phototrophs. So in the middle of 1970s, the scientists believed that the primary production was totally done by diatoms and flagellates because these two phytoplankton are the major organisms that can be found during the plankton cycle. So actually phytoplankton can be found in a lot of area, eutrophy area, oligotrophy environment. So eutrophic. Eutrophic environment actually environment that have a lot of nutrient, while oligotrophic is environment that have low nutrient. So if you read about mesotrophic environment is actually in the middle of eutrophic and oligotrophic environment. So one of the example of phytoplankton that I can give to you is dinoflagellates. Dinoflagellates is algae, as you know, zooxanthellae. So zooxanthellae is actually dinoflagellates. So zooxanthellae dinoflagellates can be found in oligotrophic environment since it has symbiotic interaction with the coral at the tropical reef. So as we know, zooxanthellae lives in the coral tissue and they have symbiotic interaction with the corals. So symbiotic interaction means that the dinoflagellate, the zooxanthellae, gives something to the coral, and the coral gives something to the zooxanthellae. So zooxanthellae use photosynthesis, undergo photosynthesis, and then it's gonna produce a nutrient. It's gonna release a nutrient. Nutrients gonna be passed to the coral cell, and in return, the coral will emit a waste product that by waste product instead of ammonia in ammonia. So the ammonia can be used by the zooxanthellae as their nutrient, as their food. So during 1970s, the plankton sampling are using plankton net that have mesh size 60 micrometer, 70 micrometer, and 100 micrometer. So during that time, the only phytoplankton can be caught by the plankton net is phytoplankton that have size bigger than 60 micrometer because the smaller one just gonna pass through the plankton net and cannot be tracked or cannot be caught by the plankton net. So recent years, the phytoplankton research already using the DNA detection and flow cytometry. So the result shows that there is a lot of smaller phytoplankton can be found in marine environment actually. One of the example that I can give is haptophytes. Haptophytes is one of the major primary producer in marine environment, but its size is around 50 micrometer since it is smaller than the mesh size of the plankton net. Most of them just gonna pass through the net and cannot be trapped, cannot be caught by the plankton net. So in the late of 1970s, the scientists have developed the observation techniques by fluorescent microscopy, and also they have upgraded their knowledge in electron microscopic observation. So when they upgrade their knowledge and also they develop the method and so on, so they have revealed that unicellular cyanobacteria is one of the phytoplankton that can be found in marine environment. The name of the cyanobacteria is Synechococcus species. So in late 1980s, the scientists developed high performance liquid chromatography (HPLC) technique. This one is a technique that been used for pigment analysis, and also they have developed the flow cytometry for cell detection. So this development makes them found a new picocyanobacteria, and the picocyanobacteria contain divinity chlorophyll A, which can absorb higher spectrum than usual Synechococcus cyanobacteria can absorb. So actually this one that contain divinity chlorophyll A, it have picobilin pigments. It does not contain this that it does not have picobilin pigments which is the accessory photosynthetic pigment that common silicococcus have. So this new picocyanobacteria was named as Prochlorococcus. Prochlorococcus has specific pigments called picocyanate. Picocyanate is actually a blue photosynthetic pigment in the cyanobacteria. So Prochlorococcus use definite chlorophyll A or B to perform the oxygenic photosynthesis, and actually they can absorb a lot of blue light in the ocean since they have the blue photosynthetic pigment. So in 1996, the scientists found cyanobacterium that using chlorophyll D to perform oxygenic photosynthesis. So chlorophyll D actually the red shifted chlorophyll that can absorb far red light, which the wavelength almost 30 nanometer longer than chlorophyll A. So the chlorophyll D actually can perform oxygenic photosynthesis using far red light, and it comes around 700 until 740 nanometer, which not all common phototroph can absorb. Cannot be used by common phototroph. Sorry for the for the sentence. So in the early year 2000, the scientists found out that aerobic and oxygenic photosynthetic bacteria contain bacterial chlorophyll A. So aerobic and oxygenic photosynthetic bacteria live in aerobic environment. And also since it is aerobic, then it needs oxygen to grow and also to undergo the photosynthesis. But since it also anoxygenic, means that it not gonna release any oxygen as their byproduct. Okay, so the phototrophic growth of some AAPB actually faster than those bacteria who are growing in heterotrophically in the dark. So growth rate of AAPB also faster in seawater other than heterotrophic bacteria. So in 2010, scientists found another chlorophyll, red-shifted chlorophyll named chlorophyll F. So chlorophyll F, we can find that in cyanobacterium that isolate from microbiome mat on the stromatolite at the coast of Australia. So actually chlorophyll F actually has an absorption minimum around 720 nanometer, and they can absorb far red light around 760 nanometer in vivo, which means that between in living organism. So in 2011, the scientists found a new cyanobacterium-like particle named UCYNA. UCYNA was detected using metagenomic analysis of nitrogen-fixing bacteria, and also UCYNA contain nitrogenous gene for nitrogen fixation. It means that UCYNA also contributing something to nitrogen cycles. Sorry. And then actually the cyanobacteria grow by oxygenic photosynthesis process. It has process like photosystem and then respiration system and also carbon fixation. But as stated in the article, UCNY, there are lacks of photosystem to rubisco, primary enzyme for fixing carbon dioxide, and also there are lack of tricarboxylic acid TCA for cycle for respiration. So this UCYNA lacking of this essential gene suggests that UCYNA might be symbiont interaction with other organisms because they are lacking of some essential which is a needed genes to be in them, but they are not there. So since it can live with the photosystem one and also with chlorophyll, so it must use it lights. Maybe it use any light energy for some activity of the particle itself. So as traditional statement that I've read in the article, so the phototroph, this is the traditional statement, which is the old statement. The phototroph, they say that the phototroph is the organism that can grow using light as their energy sources, and then the photosynthetic organism undergo photosynthesis, which is actually the organism that phototroph lah. So the primary producer, the organism that synthesize organic compound from using inorganic carbon as their carbon sources. So primary producer and phototroph are considered to be same at that time. So in current, in at the end of the article, the author have rephrased back and also has put something back in the meaning, changing the meaning and so on. So the new meaning of phototroph and primary producer are not same anymore. The phototroph are now meanings that organism that can synthesize ATP using light as their energy sources. And then the primary producer are not only organisms that produce organic compound using inorganic carbon, but also they are including the organism that can synthesize, that can produce ATP using sunlight energy. So now the phototrophs are actually equal or almost equal to the primary producer, because the meaning of primary producer are same meaning with the phototroph. So, in my opinion, the conclusion that I can get from the article is that uh, day by day, year by years, our scientists, our developer, and also our researcher improve the method, develop the knowledge, develop the instrument, develop the method, upgrading the knowledge, and so on. So by that, we can get a new things in that we cannot find in the bacteria stand. So by improving method in sampling and also in detection technique, we can find a new species. We can find a new organism that actually we don't know it exists, but with this upgrading and also development, we can see this. We can get a new information, we can get a new species, organism, and so on. So this actually when there are a new information about species, organism, and so on. So it's gonna change a lot in meaning and so on, just like the article does from the traditional understanding to the current understanding. So there is changing in the meaning of phototroph and primary producer, and same goes if we use to another field or another terms of rather than other than this one. I think that's all from me. Thank you for listening, and I'm sorry for my English, for my grammar, and also my vocabulary. I'm so sorry. So thank you.